Daniel, thank you so much for joining us. We're really excited that you could be here with us at ISHI, our 35th anniversary year. And I'd love to talk about your presentation. I'm very excited to hear about what you're going to be sharing tomorrow with the group. But before we get started, maybe you could tell us a little bit about yourself and your background.
Okay, my background, I've started my forensic career back in '92, where I joined Czech police. Became the first head of all DNA operations in Czech Republic, or Czechoslovakia and Czech Republic. After 10 years, I moved to Bosnia and Herzegovina, where I worked for ICMP, International Commission on Missing Persons, humanitarian organization mainly sponsored by the United States and Dutch government. After some 3 years doing bones from mass graves and having experience with the tens of thousands of bones, I returned back home, set up my own private laboratory, specialized on ancient bones, because the protocols we've developed together with John Daboren, they work also on ancient bones and degraded DNA. And throughout the years, I've started with animal DNA typing, and at this moment we have the biggest database of physical samples and DNA profiles of big cats, so almost 1,000 samples, and that enables us to do more research and etc. and be more powerful during cases involving big cats and other endangered species.
Yes, your case is so intriguing to me. I wish I weren't in the studio so I could come and listen to it. So you're talking about like a shocking example of the illegal wildlife trade that can happen in Europe. And the Tiger Slaughterhouse is the title. And maybe you can tell us a little bit about the case and then how DNA analysis, played a crucial part in exposing it.
Yeah. So the case, even if the first reports about the case started in July 2018. We've started our preparations 2 years earlier, because we knew that there is such a case and demand from Czech Customs and Czech Environmental Inspectorate, but we needed to be prepared to have the technology in place so that we can start analyzing the samples immediately. So the setup and everything, science behind, really within those 2 years we were able to develop some basic set of STRs, quite discriminative for big cats, and develop a quantitation system and all things together, and especially to type lots of tigers. So we had the reference samples for, so we were prepared. And when the case came, then we were able to start immediately and to process those hundreds of samples. And out of those samples that were collected by crime scene officers, we've identified some 7 different individuals, tigers, and plus a bunch of other protected species. So it was quite successful from our point of view, DNA laboratory.
Absolutely, and hundreds of samples. I mean, you're talking about quite a lot of work. What were some of the challenges in obtaining and then analyzing those samples?
Actually, it wasn't so difficult because with my background in ancient DNA and also 10 years working for police and been involved in many research projects. So it wasn't so difficult. So working with tiny amounts of DNA. It's my daily bread, so really. But there are other cases where we have to do some additional research. For instance, cases involving tanned skin or fur, because then mitochondrial DNA can be altered by the chemicals used for tanning. So then it becomes more challenging, and that's what I love, because I love obstacles, so I can climb up.
That's very interesting how that would change it. Yeah, how do you overcome some of those obstacles when that happens?
I'm using my scientific background and I'm talking to my colleagues and also being ex-police officer, I know that it's not only DNA, but we also have other disciplines I can use to confirm our results. So like with FUR, I can use hair morphology and etc., or some proteomics, and I know the ropes and what to do, to have my results bulletproof. So.
That's perfect. That's amazing. I'm sure that you come across highly endangered species in your work. And does that differ in how you analyze that? Does that make it more challenging?
It's a very good question, actually, because with animals, you're dealing with different spectra of samples. Like the first case we were involved was included rhino. And rhino skin is extremely, hard, to sample and how to work with it. And suddenly, we needed to process feathers and some traditional Chinese medicine, some tinctures, and so what to do and how to proceed. And suddenly we started to validate and new extraction methods and etc., especially removal of inhibitors, because suddenly we were facing completely different spectra of inhibitors when compared to human DNA identification. And that's what I love on it, because then you have topics for students and everything for my presentations, and so I'm not just doing the same boring stuff day after another, but we really on our laboratory meetings we are solving problems and like last case. Human contamination, so we've developed suppressors so we can amplify just animal DNA, any animal, but not human DNA. So it helps us to analyze more samples in a more fluent way.
So that's very interesting mixture. It's a little bit different, but
it's flipped over because normally
right, different mixture analysis.
But even this human DNA can link the perpetrator to this sample. So, but yeah, we can do both. So,
okay.
But for species identification, we need to have unique, special approaches and.
Speaking of that was actually my next question. When it's species specific, what kind of innovations have there been or that you're working on that help in that area?
Species identification. I would say within those. Past 10 years, the technology has evolved significantly, and especially, we have specialized databases like BOLT systems, and it's, targeting cytochrome oxidase 1 gene, mitochondrial gene. So, and they are verifying the records in the database, so it's much more precise. But me being, ex-police officer, I have to repeat it. So I know that. I must really deliver the perfect product. So we are also using other databases. We have our own internal database. So I'm everything checking twice or 3 times just to be sure it fits and I'm not releasing falsely positive or falsely negative results.
I think that's really interesting to bring all of the experience you have as an ex-police officer to the work that you're doing now, because you really understand and speak the same language as you're working together.
Exactly. Yeah. And I think my biggest advantage is when I go to the court to defend our results with 30+ years experience, and I'm going to the court on a daily basis, and even when I was working in Bosnia, I've been doing reports for International Court Tribunal for former Yugoslavia, so I'm quite experienced, and it helps. It really helps, because I'm also teaching at the university for some 30 years. I like the interaction with students, so I'm able to explain things in a simple way, but on the other hand, when the other side tries to somehow crucify me, I can, do the same to them in a very effective way, because
it's very helpful. We actually talk about that a lot, because there's a lot of education usually so people can understand the other side, but you naturally do because you've already been there. What impact do you hope that, in this case particularly, what impact do you hope that it will have going forward with the illegal animal trade?
The reason I've decided to to come to United States to talk about this case and our identification efforts is that it's still not the topic number one of non-specialized conferences like you have for wild animals. And when I've checked the spectra of presentations on issue, it's mainly genetic genealogy and some rapid DNA and etc. But okay, I understand it because those guys are working on this on a daily basis. But there are special cases, and they should know what to do, and maybe also it can force them to start thinking in slightly different ways what can be done, not just to buy kits, but we are developing all our kits because there is no manufacturer that would prepare a kit for tigers or other big cats or some parrots. So we have to do everything ourselves. But suddenly with my experience, 30 years ago, we were also doing the same, but without the technology in behind. So now we can do everything much faster. We have draft genomes and everything, and it's much easier. Really, the life is much easier. And within those 2 years, we've really prepared complete identification kits, quantitation, STR typing, including color letters. Yeah, it can be done because once you know how proceed and what to do, it's much easier.
I think it's fascinating because there is like a cross-pollination of learning then too. I mean, you're right, certain topics are trending, and so we see a lot of that, rapid DNA, IgG, things like that.
And I remember, those 20 years ago, I've been, reporting about mass graves and, bone samples, and I've been organizing, bone workshops all around the globe, and just teaching people how to extract DNA out of bones and how to use it, and with a big help of Bruce Badavli and others. And suddenly, okay, it's no longer emerging topic, and everybody's using it. So it's okay now. It's genetic genealogy, but I've been using this investigative genetic genealogy 15 years ago. So it's nothing new for me. I just wasn't talking about it. But maybe, and I hope that this topic then and my presentation tomorrow will bring more attention, and maybe Promega will invite me to run a workshop next year in Florida. So yeah, that's what I hope for, because it can be really interesting for others, because I can see it. My students, I start with some basics about forensic genetics, history, etc., and normally on the first lecture you have 100 students and the number goes down. But when I have a lecture about animal DNA typing, because I always talk about cases I work on, and it's back again. And I have even more than those 100 because they bring their colleagues and friends, and so I can see it's also for them it's fascinating, and there are lots and lots of students that would like to work in my laboratory doing their diploma thesis, but unfortunately having a private lab without any external governmental sources of money, I can take one or two. But the best students from my class, that's good because I can see how they interact. So it's some pre-selection. So and it helps me.
Well, I was very excited. I remember when we were looking at the abstracts, and I was like, absolutely, like we need to have animal. I think the last interview I did that related to animal was, illegal hunting in Canada, and so they were trying to tie it back to whoever. So I was incredibly fascinated by this. I'm so happy you're willing to come over and share it. It's going to be a great presentation. And I think another.
It was my honor.
Yeah. I think another interesting part about it is DNA barcoding methods. Can you talk about that? Some of the, you've been using that, and the advantages, limitations.
Yeah. We have to distinguish, human identification, individual identification of animals. It's the same. So we can do, parentage testing, some smuggled animals, and etc. But then it's quite easy. But then you have cases where the customs sees some artifacts, storage Chinese medicine, and they think, oh, okay, what about if those Vietnamese, Chinese are trying to smuggle some products, of endangered species, to Asia, and that's my task, to say, okay, it's clean, that's just some pork meat in it, or yeah, they've used tiger and this and this and that. And with the broad spectrum, because those are samples you wouldn't expect, can contain any, biological material. So we had to develop many assays to be able to answer the questions in a timely manner. Really, let's say within a few days to say, okay. But I know it's not enough, and that's why we are even working on some, I call it a point of care or, point of action, small instruments that bring you the answers in tens of minutes, and imagine that at the Prague airport they were for the whole month, I think it was back in 2016, searching our luggages for presence of biological material, and sending this stuff to my laboratory. So it was, a huge amount of samples and everything. A lot of paperwork has been connected, but we really identified many endangered species in those, tinctures, and other stuff has been seized, and it was amazing, how effective it is when you do, a very proper searches, sniffing dogs and everything, and all the technology that's available at this moment. So, but the workload was so high that even for the customs and inspectorate, that it was just 1 month, and it was the shortest one. It was February, and then the way to process everything, till the next year, we've repeated the same, and again, there are many cases, and
yeah. And it's amazing to me.
Sorry for interrupting you, but
no, please.
Imagine that they take tiger bones and they boil them for 7 days and 7 nights, so 7 times 24 hours of this quite extreme heat, and then they prepare something that we call a solid broth, and they cut it, and then they use it as some traditional Chinese medicine, but imagine that even there is some tiny amounts of proteins and DNA that can be used to say, okay, there's tiger inside. So 4 years ago, I believed that such a long heat would destroy DNA, but when we did our experiments, we see, okay, it's typeable. We can do it.
So that, really, that's like, talk about degraded DNA, or, DNA that's challenging, but you were able to do it. And it almost seems like, correct me, I'm probably going to say this wrong, but yes, you're kind of coming up with your own rapid solution, when it's found in a suitcase, and trying to help speed that up. So it's effective, and you can trace it back.
We are trying to help, and I really love the interaction with guys from customs and even police forces, because I need to know what's necessary to be done, because me as a scientist, I need inputs, what's necessary to be done. So and once I have this piece of information, then it's much easier.
Yeah. So this all seems relatively new, and we talked a little bit about the logistics of it, but what else would you like people to know about what's happening now and how they might apply it in other areas?
One important thing is that there are many regulations regarding biological materials. Shipping biological material overseas, but even from continental Europe to England, it's very difficult, and many limitations, and you must have so-called permits, and so that's why I would like to see specialized laboratories on every continent, but I do not think it's possible, because there is this question, who will finance it, and but I think that this illegal wildlife trade is the third most lucrative for different mafias and smugglers. So I think it's worth to cut those black money. But it's
Yes,
a question for our authorities. But I'm really free to share the knowledge and we have and everything with others because. It's very difficult to make profit on it because it's not human. For tigers, maybe several hundred cases per year. It's nothing for big pharma companies, but for small entities, universities, it can be interesting.
Yeah, I think it's
To
incredibly
do some
interesting.
casework and combine it with some research.
Yeah, not everything should be driven by a profit motive. I mean, there's an ethical consideration and trying to stop this in other areas. With the variety of species, does that, if you were going to be able to expand this, across different continents, how do the variety of species affect that, or would it, would it be, fairly simple to?
I would say that every region has its, own species that are, smuggled, and let's say out of Africa, ivory. That's definitely number one, and you have laboratory in Washington D.C. It's specialized on ivory, some visor. Then regarding rhinos, it's in South Africa. Cindy Harper doing an excellent research, individual identifications of rhinos, and also building a database so that you can really trace, rhinos to the original, farm or reserve. And surprisingly, my small laboratory in Prague is the reference laboratory for tigers and other big cats. And it's because, Vietnamese started, with their, illegal trade with those tigers. Because, that tiger is much more valuable than the living one. And you can convert it to thousands and thousands of euros or dollars. Mm-hmm. And that's why they do this when... They can make some money out of it. They do it. It's like marijuana or drugs, and they do it. Yeah. So that's why, and we have the highest population of Vietnamese in Czech Republic, and that's why we have so many cases involving big cats and.
It's amazing that forensic analysis can help solve this problem and really be an integral part of it. Something we couldn't have imagined, decades ago. Yeah.
But I've sneaked out of your question, regarding those new technologies for species identification. So, when we talk about Sanger sequencing, I would say it's a golden standard, but not always, it's so easy because when you have a single source, so just a tiger and some medicine, it's okay. But you can have more animals, then it's a problem. So you have to use some massively parallel sequencing, and it's okay. Fortunately, we have tools that we can use. But we've started to research also another possibilities, and just recently published a paper on length polymorphism on mtDNA in a control region. And so it's also suitable, and there is a similar approach called Spindle. It's a group from Portugal. So there are also other approaches, and technologically, it's we would skip back to the early 90s, using mini satellites and etc. But with the knowledge of past technologies, we can do it. But we are also using, for instance, high resolution melting analysis. And some novel one, the LAMP assay, so isothermal amplification, and again we are trying to develop something that can be used in the field. So that's what we do and how we play.
Creative approaches, I love it, so that you can tailor it. Certainly gets more complex as you add different species and regions. But where do you see your research going in the future? What's next?
It depends on research grants
of course,
we receive, because we are secure till the end of the next year, And what's gonna be next, I don't know. And but yeah, having this very valuable database of pickets, I think we will find some sort of financing and some support, even if it's not from our government, can be from abroad. So.
Yeah. Well, I love that you're able to share that here, and really appreciate you doing the panel, but also sitting down with us so that the video can reach other people who may not be able to attend, but would like to know more about this. Yeah. So I think it's fascinating. I can't wait to learn more about it. Is there anything that we didn't touch on that you'd really like to make sure we include in the video?
Actually, the list of your questions was great. So I think we've covered all areas that are important.
Well, thank you so much. And how are you finding Ishi so far? It's our 35th anniversary, so we're checking in with everybody.
It's different if I compare it to other conferences, like your main competitor, ISFG or
2 weeks ago,
European Academy of Forensic Sciences. And what I love is are those small differences. For instance, we've been exhibiting once at ISFG, and nobody came to thank us, distributors, and so it's not fair because they can organize the conference because they are non-profit out of our money, company money, but it's completely different, Okay, it's a perfect setup because you have posters, booth, everything, food served in the same area. So because you're a company that knows how it should be done, and also the feedback. So you're trying to improve your next and next and next, so to deliver better and better products. So that's what I love. So.
Well, we are very lucky that you were willing to come and be a part of it, and we definitely
thank you. And I promise
I
will do my best to come next year to Florida.
That would be fantastic. I hope we get to speak again. I would love to hear, what's next and what's happened over the last year.
And I will bring some new cases with me.
That would be fantastic. Please do. Okay. Thank you so much, Daniel. We really appreciate it.